Okay, we are close to 12:05, so I think we can get started. So thank you everyone for joining this month's Grundfest Memorial Lecture. Before we get started, just a reminder that we usually begin at 12:05 and end a bit before 1 p.m. And if you want to keep up with future talks in this series, you can visit the website that's on this slide. This lecture series is hosted in remembrance of Professor Warren S. Grundfest, who was a professor of bioengineering, electrical engineering, and surgery at UCLA. Professor Grundfest was a SPIE fellow in biophotonics and also a pioneer in many imaging modalities. He was also a strong advocate for junior scholars, which has inspired this lecture series and its focus on highlighting early career researchers in computational imaging and related fields. Before we continue, I also want to thank the co-organizers of this lecture series, Professor Achyuta Kadambi at UCLA, Professor Katie Bauman at Caltech, and myself and Pradyumna Chari, who are PhD students at UCLA. Finally, I want to thank the sponsor of this lecture series, Akasha Imaging, which is a computational imaging startup on polarization. Akasha sponsors an inverse honorarium for speakers in non-permanent positions, such as PhD students and postdocs. So thank you again to our sponsor. And with all that, it is my pleasure to introduce this month's speaker, Regina Eckert, who is a postdoctoral fellow at the Jet Propulsion Laboratories, JPL. Currently, Regina works on computational imaging algorithms for remote sensing imaging spectroscopy, and the work, the method she develops will be deployed to NASA's airborne instruments in the future. And Regina completed her PhD recently at UC Berkeley with Professor Laura Waller, where she worked on computational imaging systems for high resolution and high dimensional microscopy. So I think today Regina will tell us about some of her recent work. So I will hand it over to Regina to tell us some more.
All right, thank you so much, Ellen, for the introduction. Let me share my screen. All right, just to verify, is everyone seeing my slides? All right, cool. So today I'm going to be talking to you about computational imaging from chloroplasts to tree canopies. This is slightly tongue-in-cheek because today I'm going to be talking to you about kind of two separate pieces of work that I've done in the past. One where we're looking at 3D quantitative phase imaging with some applications, including imaging of chloroplasts. And then also work that I'm doing at JPL, where we are working with remote sensing and hyperspectral imaging, where we are retrieving surface reflectance of things, including tree canopies. And so I just kind of wanted to think about, computational imaging is something that we apply across all of these different scales. And how do we actually kind of communicate amongst those scales and applications? How do we actually translate different methodologies amongst those things? So that's kind of one of the higher level things, but also dig into the guts of these projects. All right, so computational imaging from 40,000 feet. This might be one way that we look at it, where we're literally at 40,000 feet and we're doing some imaging. We could think about also what's computational imaging from a submicron view. That might be this microscope where we're actually looking at a submicron view. But I think, maybe a little bit less literal interpretation. If we're looking at high level computational imaging, I think what we all really think about is kind of some imaging problem where we have some object that we want to understand and get some relevant information about. And we have a sensing system and a processing algorithm, or mini processing algorithms, that we're trying to optimize, right? This is, I think, something we've all maybe seen in many different forms. Kind of a definition of computational imaging is this end-to-end system where we're trying to get something useful out, and then we're optimizing this whole system together as part of that. And throughout grad school, like I saw this many times, I put this in my slides many times. I think it's a really exciting idea to think about kind of co-optimizing the sensing system and the processing algorithm. But what I was experiencing on the ground in graduate school and also now as a postdoc is something that's a little bit different, right? When we're looking at a computational imaging system, sure, we're still looking at kind of an application space and retrieved information. But when we're zooming in on all of these things, there's just like tons of questions about every single aspect of it. And it becomes very difficult to think about that high level co-optimization of optics and computation. So there's different questions that we might even ask about, what's my application space? What's the useful retrieved information? So here I'm showing this cluster of cells, but what type of cluster of cells? Is it something that needs to generalize across cells? Or instead are we looking at trees versus grasslands, for example? Retrieved information could be an actual reconstruction of that object, or it could just be a classification. Is this cancer? Is it not cancer? That type of thing. Then we get to all of the kind of specifics of both the physical system and the alignment. What type of optics am I using? Where is it going to go? Is it on a microscope that's on a bench? Does it have to be transportable? Things like that. What type of physical model am I using for how my light is propagating through that object or interacting with my imaging system? What algorithm am I using? What loss function? And then, beyond that, at least personally, the majority of my time in graduate school was actually spent on model mismatch and eliminating model mismatch and calibrating my system, as well as validating and communicating how reliable my results were to people. So that's maybe like the kind of really in your face, like all of the details of something that it takes to put together an imaging system. And this isn't even everything, right? We also have time to design and to produce and also to use that system, the money it takes to do all of those things as well. And then also, is it usable and is it equitable and ethical? Like there's just tons of different questions when we go down to like a very focused view of what an imaging system is. It has a ton of these parts. So I know personally, I was kind of in graduate school looking at like, well, here's this really high level thing, and I think it's a really great idea. But then when I'm actually in the weeds doing the work, how do those two things relate? So this presentation is going to be an attempt to kind of draw some meaning from two different projects that I worked on. But I think this is still a question that I'm asking, and I think a lot of people are asking this question because you listen to a lot of talks about very specific kind of goals and applications and ways that those were approached in different areas, and you think, how can that apply to my specific problem? So that's just a question that I'm trying to ask, and we'll see what progress we can make on it here. So again, today I'm going to be talking about two different projects. One I'm labeling chloroplasts, so this is a submicron view in the strictest sense, so it's microscopy. And then we'll be talking about tree canopies or remote sensing. All right. And if at any point you have a question, please just jump in. We'll also take questions at the end, but happy to take questions in the middle. All right. So for this chloroplast section, microscopy, we're going to be talking about 3D quantitative phase imaging, specifically kind of using this measurement diversity thought to make it better. All right, so let's start out with what is quantitative phase imaging? So in quantitative phase imaging, we're using the actual delay of the light through our object. So here we have this cluster of simulated cells again, to understand its properties. So we're measuring this light's phase delay, and that is related to the refractive index of the object, so how that material changes the speed of light. And that in turn is related to different properties of the organelles, the nucleus, things like that. And it really gives us a lot of good structural information for different types of biological cells. So that's why we're doing this. If we wanted to approach this problem, we have these transparent cells. How do we actually get information out about them? So we'd want to kind of model our object here, X, and we want to illuminate it with something. So we're going to choose coherent illumination because we're trying to measure phase. So coherent illumination makes sense. And then that's going to scatter through our object in some modeled propagation way. and we're going to call that an operator S on our object. Then we'd better image it. So we're going to put a lens there, and it's going to be an objective lens because it's really small. And that's going to do a Fourier transform. And it's also going to filter our light with a pupil function in the Fourier plane. Then we're going to use a tube lens to refocus it so that we can take an image at our camera. And of course, our camera can only measure absolute value of our electric field. And so that's what we're actually going to measure. All right, cool. We got a nice imaging system. Now I can just throw it into an optimization problem and solve, where I'm kind of comparing my measured image Y to this modeled image, and I can update my object X in order to get a good kind of matchup there. And we're happy, we're good, we've solved it. Obviously not at all. So we really need a lot more measurements than just one in order to get 3D refractive index. So we can take this kind of basic setup here that we have this illumination and all of this modeling that we've done and try to figure out how do we actually measure good information to get 3D refractive index. So the way that people have done this in the past is you can change the illumination angle, and that's going to be similar to kind of like a computed tomography where you kind of have a CT scan, that is, where you're changing your illumination. It's not quite the same, but it's similar. You can change focus. So that would make sense pretty natively for 3D information. You're trying to get something that's kind of depth information. So you want to change your focus and see what parts are in focus and what aren't at different depths. And you can rotate the object. So this is related to the change in the illumination angle, slightly different. All right, so those are different ways that people have gotten 3D information in the past. What I did in my PhD was I said, okay, well, how can we actually encode this information about this object kind of best in our measurements that we're taking? And the system I came up with is to combine this kind of change in illumination angle thought with the thought that we want to change our focus for different measurements. And what this culminated into, what it kind of ended up being, is a change in illumination angle and coding at our pupil plane. So we're kind of introducing this spatial light modulator element into our system to try to constrain our measurements more. Something else that you can do with this that I'm not going to talk about in this talk, but you can also use this in an end-to-end learning situation and learn better pupil coding designs, which is fun. So, but just to go over the method itself a little bit more in depth. So here we are again combining this illumination side coding with our angle scanning mirror. So we're having a plane wave that's going to have different illumination angles into our object with some detection side coding. So here we have our conjugate Fourier transform planes. So we're relaying the pupil plane of our object onto a phase spatial light modulator. And then we're going to image it. And on this phase spatial light modulator, we can have different kind of just programmably different pupil codes. So we could try defocus, which would be strictly, again, that idea of combining illumination change with focus change. Or we could do something completely random, like random pixels. Or we could go somewhere in between and do, say, random weights on a Zernike basis. You can basically do anything here. These are just three things that we tried. There's kind of a lot of work that then went into which one is best. Long story short, they're all pretty much the same, which is very interesting in itself. But the Zernike ones are fun to look at, so I'm going to show you results from that. And all of this work here that I'm going to show in a second is from simulation, but we also tested this in experiment, and I'll show that at the end. All right, so the basic idea of doing this kind of dual illumination and detection side coding is that we're actually increasing the measurement diversity in our system. So what do I mean by measurement diversity? At least the thing that I kind of like to think about is what is the diverse information content in my measurements? So that's kind of like, yeah, that's a cool idea to think about, but what does that actually look like? What does it mean? So here we have our simulated cell cluster, and we're going to simulate some measurements and just look at them and see what measurement diversity might mean. So in this case, we have different angles of illumination going down in the different rows. And here we're showing images where we don't have any pupil coating. This is what these images would look like out of a kind of normal, just you're changing your illumination angle type of system. And what we're seeing here is that this 3D cell cluster, there's some changes between these angles of illumination. So we're seeing out of focus things kind of shift around. There's a little bit of different contrast. But if we include the Zernike basis coating, we're actually getting way more physical contrast between our images. And so we can like kind of physically see that we're encoding different information across these images than we would otherwise. All right. So we're going to kind of use this system that I've just proposed. And again, we're going to reconstruct with an optimization algorithm, very similar to the one that I showed before, where here our system model is including our angled illumination, our multi-slice forward model, our objective pupil, and now our pupil coating as well. We're comparing to our measured images, and we're going to reconstruct some volume. So the volume kind of shorthand here is that we have on top an XY view of our cell cluster, and on bottom an XZ view. All right. So looking at this system, if we give both the no pupil coating case and this Zernike coating case enough time and enough data, they both converge pretty well. And that's comparing to this NA filtered ground truth, where we filtered by the actual kind of information collecting ability of the NA of our objective lens. So what I mean when I say they both converge is that the cell backgrounds, so these low spatial frequencies, are both recovered fairly well for both of these methods. But if we limit our illumination angles, limit our computation time, or increase our background noise, we actually do find that the pupil coating is much more robust. So we'll look here at this limited illumination angle case. So this over here is a little bit of shorthand, so I apologize for that. But basically what you're looking at in the blue is the angle of the illumination. And then in the red is the NA, or the numerical aperture of our objective. And so what we're seeing is that when we fill that NA of our objective with our illumination angles, we're doing well in that previous case. And now we're going to limit it down so we don't need as high of angles on our illumination side. And not only is this a data reduction, but it also would make our system a lot easier to align and to design, because now we would no longer need as high of a numerical aperture or as high of an angle input into our system on the illumination side. All right, so when we look at the reconstructions in this case, we are now seeing that when we have no pupil coating, we lose a lot of our low spatial frequencies in our phase reconstruction. But when we have our Zernike coating, it's actually doing pretty much just as well as it did before, even with a lot less data and a lot less kind of high illumination angle data. So that's really cool. I think it's just exciting that this actually provides something that's useful rather than just being like, oh, that should be useful, right? All right, so you might be asking, does that work in experiment? So we have a 3D printed refractive index phantom that we tried this on. And indeed, we see that when we reduce our number of images into our reconstruction algorithm and reduce the max illumination angle, we're still seeing better low frequency reconstructions with the Zernike coating than when we don't have any pupil coating. And that kind of holds true as we decrease the number of images. So the reason this happens is because this pupil coating actually introduces better low spatial frequency weighting. So there's a lot here. We can talk about this more if people want to, but just basically to say what you're looking at here is that as we limit the illumination angle, the spatial frequencies that are represented in the kind of weighting of the different measurements is reduced when we don't have coating, but stays fairly high when we do have this pupil coating element. Which means that this kind of diverse pupil coating couples varied phase information into our intensity measurements. This also makes sense from kind of a physical perspective. So we have our object, we have this coherent light propagating through it, And if we don't have the pupil coating element and we just measure at that point, then we're not going to get any more phase information than is already included in that kind of varied illumination angle. But if we kind of can modulate the phase, then we're going to have a different measurement that we're measuring in amplitude at our camera, and that's going to include more phase information. All right, so you can look at regular biological samples, you can look at nice RI phantoms for this, but one fun application is looking at chloroplasts. So here we have these chloroplasts that are kind of scattered in 3D. And the people who were wanting to look at this work were trying to understand the three-dimensional structure and how the chloroplasts changed with exposure to light. So we did a little bit of imaging and could see some fairly cool kind of like structural features on the cell, including in 3D. So you can kind of see the striping here in the Z plane on the bottom, which is really cool. So. All right, so that's our whirlwind tour through 3D RI imaging. Next, I'm going to be talking about surface reflectance retrievals with spatially constrained atmospheres. But I'll pause, and if there were any questions first about 3D RI, though again, you can ask them later as well. All right, not hearing any unmutes, so I will continue. All right, so cool, microscopes. Microscopes are great. But, you know, if we're trying to look at big things, just completely different application space, let's look at the Earth instead of the cells. We have kind of some different application problems, different things to work through. So here's this application space. Instead of looking at cells, we're going to be looking at the surface of the Earth. And we're going to be doing it with visible through shortwave infrared imaging. So JPL, this is work done at JPL, produces some really high quality imaging spectrometers that are push boom spectrometers that measure both the kind of wavelength visible through shortwave infrared and a single spatial dimension. And then, you know, it's mounted on a satellite or a plane. And so it's naturally going to be moving through that other spatial dimension. And so it's a push boom spectrometer that just builds up your image cube through the motion of the imager through space. And what this provides is that every location on this image has a high quality spectrum that we can look at. So this is a spectrum from the EMIT instrument, which is currently on the ISS. And these imaging spectrometers are actually used across the solar system. So some of these have not actually launched yet. Some of them are already in place. But planetary bodies that they're going to be going to are Europa, the Moon, and Mars. And as well, of course, as our lovely, lovely Earth that we love to inhabit. So one of the useful cases of this is we can take this spectrum and we can identify some mineral absorption feature and we can detect minerals with it. However, it's also useful for kind of a wide variety of applications. So snow and ice, monitoring oceans, vegetation, dust plumes. There's lots of different applications if you can both get the spatial kind of distribution of information as well as this spectral component. However, one lovely thing about the Earth, but also a slightly pesky thing about the Earth, is that it has an atmosphere. So having an atmosphere actually obviously enables us to do all of the great things that we want to study. So it enables having vegetation at all. But the problem is it also introduces lots of kind of scattering and absorption in our spectroscopy that is not useful for understanding what's happening on the actual surface of the Earth. So in order to get something useful for those applications I was just talking about, you have to do an optimization problem where you get the radiance that was actually measured at your sensor, and you retrieve from that some surface reflectance values, as well as something about your atmosphere. So in this case, water vapor or aerosol optical depth. And the problem that a lot of us at JPL are looking at is how can we actually improve this retrieval process? It's doing really great as it is, but there's lots of ways that it could also be improved, right? So I'm going to talk a little bit about some work to introduce spatial information into this process. Because of course, here we are in this imaging community. You have something that's an imaging spectrometer. Why would you not include spatial information? Let's do it. All right. Just to set up what you'll be looking at in the next few slides, this kind of forward simulation process I have here is we have this surface reflectance cube. That's our ground truth. So I'm just showing a false color RGB, but there's actually like 425 spectral channels there. We have an atmosphere. So we have this water vapor component, and then also this aerosol optical depth that we're going to simulate. We use our ModTran forward model to simulate what radiance we would get at, an aircraft level. And then from that, we can reconstruct, obviously, via an optimal estimation algorithm to get that reflectance, also the water vapor and the AOD. So that's the general process that we'll be looking at here. All right. So the interesting thing that I found when I joined this group is that currently a lot of these reflectance retrievals are done on a spatially independent basis. So they actually treat every pixel independently and just look at the spectral component. And for each spectrum, do this inversion process where you do this optimal estimation to estimate what's the water vapor, what's the AOD, and therefore what's my reflectance. And as you might expect, the problem with this is that you end up with a lot of spatial surface biases because you're not considering that spatial aspect at all. So you can kind of see that here in this retrieval error map. You can see kind of different roads and different surface features showing up in our atmosphere. And then those are also propagating to error in our actual reflectance cube. This makes a lot of sense because if you have a road on the Earth, the algorithm here is going to estimate that independently, and it's maybe harder to get those atmospheric values over things like rocks and roads. But it doesn't actually make sense that your atmosphere would be any different over a 10-meter patch of road versus the vegetation on either side of it, because you're looking at the whole column of the atmosphere above that piece of ground, not just kind of local changes in water vapor. All right. So the question I was kind of asking was, how can we address this problem? And the hypothesis here was that if we leverage the spatial smoothness of the atmosphere, so using that information that we should actually have a spatially smooth atmosphere, we can improve these retrieval errors. Right. So the proposal here is that in order to constrain this atmospheric spatial smoothness, we'll follow this kind of new algorithm where we have our radiance, we cluster it into superpixels. Then we do optimal estimation on averages over those superpixels. And this is to get kind of an initial atmospheric state that we then put through a Gaussian process regression to estimate a smooth underlying atmosphere. From that, we can then retrieve our surface reflectance using optimal estimation on every pixel, but fixing our atmosphere. So this is kind of our process that we're going through, and then the results that you'll be looking at is these kind of Gaussian process constrained spatially smooth atmospheres, as well as this recovered reflectance. So I think the really cool thing about that also, it makes like intuitive sense to me, but I think what's really cool too is that you can actually show that's mathematically equivalent to simultaneously retrieving a spatial and spectral retrieval, as well as being computationally tractable. So I think that's a big statement, right? So I'm going to walk through it a little bit. So beware, math is coming. Not that you all are afraid of that. So, all right. So again, we have this traditional pixel by pixel optimal estimation algorithm. What that looks like is we have our forward model where we're measuring our measurements y of radiance. We have a radiative transfer model on our atmosphere and our surface, and then we have some measurement noise. And what we're trying to do is this optimal estimation algorithm, which is also actually just a map estimate, so a maximum posterior estimate. So we're trying to maximize the probability of our surface and our atmosphere reconstruction given the measurement y at that pixel. To do this, we're going to be minimizing this cost function, which looks very ugly, but is actually pretty straightforward. So we have our data fidelity term that we're trying to minimize, as well as our priors on S, A and S. So we're going to introduce some priors on the covariance there. All right. So that's our kind of traditional algorithm that we're going to use. Now we're going to introduce this new constraint that our atmosphere is spatially smooth. So we're going to introduce this joint prior distribution over all of our atmospheric parameters across the image to account for those spatial correlations. And we're going to decide to use a Gaussian process prior because that's efficient to calculate. That doesn't necessarily mean it's what you have to do, but you can. There's many ways you could implement this. And when we do this, when we have this joint prior distribution that we introduce, our surface reflectance and our measurement each become conditionally independent of spatial location once our atmosphere is known. So then when those are conditionally independent, we can use belief propagation methods to calculate the marginal distributions of all of our variables. All right, there's a lot of words here. One way to break this down here is, again, we're trying to now maximize our surface reconstruction given all of our measurements Y across the image. And we're going to use the message passing of belief propagation. This can look like this factor graph of marginal distributions, where each of these rows is related to the Gaussian process prior, the radius transfer model, or the surface prior distribution. And you can use this factor graph to kind of construct what my probability of my surface measurement S is as a combination of all of these factors. So that's one way to look at this. The way that I think is a little bit maybe more easy to look at is here. So I've again just kind of put that same equation that we were just looking at again, but now I'm going to break it down in a slightly different way. So here we have our initial solution for our atmosphere with an initial prior. Then we are estimating a smooth atmosphere with this Gaussian process regression that's relating all of our atmospheric parameters to each other spatially. And then we're retrieving our surface reflectance prior with a new prior, S on S. So what you might notice here is that this is exactly what I was showing you before. We have this kind of initial solution. We're estimating a smooth atmosphere with the Gaussian process regression, and then we're retrieving a final reflectance. So that's the relation to the math underpinnings here, which makes this a lot more rigorous than just kind of some ad hoc algorithm that might work. So that's very exciting. The question is, does it actually work? All right, so we again have our ground truth and our pixel by pixel retrieval, which I showed you before. And now we're going to look at this spatially constrained retrieval. So what you might notice here is things like this road are showing up in our pixel by pixel retrieval, but not in our spatially constrained retrieval. And these trees do seem to have an effect in both actually, but on a lower scale for the spatially constrained work. This is easier to see when we look at the retrieval error. So again, we can see lots of surface features showing up when we don't include this spatial constraint, but not when we do include that spatial constraint. You will notice though, obviously there's not everything is being captured by the Gaussian process prior. There could be different kind of kernels that you could use to make that better. But the general kind of idea here, I think, holds true that by introducing a spatial constraint, you're going to kind of make your system reflect what's actually happening in the Earth a lot better, and then you're going to improve your results. And the cool thing is that this isn't just spatially, we're also seeing a spectral kind of improvement. And so you can see that the spatially constrained method is getting a lower error across the spectrum compared to that pixel by pixel method. The other cool thing is that if we kind of ramp up our simulated noise, so those were all results shown with kind of a typical noise parameter from our Avarice NG spectrometer. That's an airborne spectrometer that's been used a lot over the last 10 years. If we change in our simulation the noise values that we're using in our radiance, we can see that not only does this spatially constrained method have lower error overall than the pixel by pixel method for the reflectance, but it's actually having lower error for the atmospheric values as well. So basically what's going on here is this pixel by pixel element is using the water vapor and the AOD as these kind of like nuisance parameters where it's just throwing error into them and maybe not necessarily retrieving them accurately in order to deal with noise. But when you actually have this spatially constraint, you can properly allocate the noise a lot better. Right. So what I've shown here is this spatially constrained retrieval method, which reduces surface biases and is more resilient to noise, and is also mathematically equivalent to this kind of spatial spectral retrieval. All right. Pause for any questions about remote sensing. All right, cool. So now we're going to go back to what I was asking at the beginning. How do these things actually relate to each other? I just showed you a lot of kind of work on two very different projects. How do you parse what's useful out of that, right? So we're going to return again to this kind of computational imaging from 40,000 feet, where kind of ideally we're taking this sensing system and we're able to change it easily. And we have these processing algorithms that we're also able to kind of change and optimize easily. We're going to optimize between the two of them, which I think kind of is our goal for a lot of computational imaging things. And the interesting thing when I was kind of reflecting on this work is actually in this talk, I've played a bit of a trick on you because I have my 3D QPI system and I pretty much fixed that processing algorithm and did a lot of optimization on the sensing system. And then in the spectroscopy case, the sensing system is pretty fixed just because it takes a long time to create. And also there's imaging systems that have been flying for 50 years out of this group, so they literally don't change. And what's being updated is this processing algorithm that's going to be dealing with it. So, okay, that's interesting. How does that fit into this kind of overall idea of computational imaging? And the thing that I think anyway, or at least one answer to this question, I think there's probably many answers to this question of how these things relate, is really thinking about, okay, for computational imaging, maybe rather than thinking about this co-optimization problem, really thinking about when I'm imaging something, in what dimensions can I actually encode useful information? And then also in what dimensions can I extract useful information? So kind of really centering that measured data at the center of this system and thinking about that as the kind of the prime thing that we're both trying to encode more information and get more information out of. And what are the dimensions on both sides of that we can do that? Which really is maybe in the end exactly the same thing as saying you're going to co-optimize these two systems, but is maybe a different way to think about it, and is definitely a way that I've found useful when trying to bring new ideas and fuse new ideas into these different types of applications. So just one thought. I'm curious to hear what other people's thoughts are on that, because definitely it's so interesting kind of watching different talks and seeing what other people are working on and trying to understand how does that relate to this specific thing that I'm working on. So, all right. So with that, I want to acknowledge both of the groups that I've done this work in. So the Berkeley Lab and all of the Berkeley folks and other collaborators that were involved in this work. As well as all of the folks I've been working with at JPL this year. Just big shout out to all of them. Obviously, all of this is a team effort. And also thanks again to the organizers for letting me come talk. I also have some kind of lessons learned as requested from grad school, but I think first we'll open it up for any technical questions. So thank you all so much.
Thank you for the talk. Let's give a quick virtual round of applause for Regina. And if you have any questions, feel free to unmute yourself or drop them in the chat and I'll read them out.
Yeah, I have two questions. So the first one is about the face image. So for solving that optimization problem, so what algorithm do you use?
Yeah, so we were using a FISTA-based algorithm, and it was basically kind of going through each image and doing kind of gradient descent, basically. Yeah.
Okay, okay, I see. Oh, about the remote sensing imaging, is the transformation model, for example, this FAS? Is there an analytic form, or is it just some abstract operator there?
For this one?
Oh yes, about this FAS. So do we have an analytic form for it?
No. Well, yes, actually. So it's kind of complicated. So we do have, there's like a relationship between like transmittance through the atmosphere and your measurements that you can write out, but you have to like actually simulate through a radiative transfer model to get the transmittance and other things like that. So there's been a lot of work on building emulators and lookup tables and things like that to make that a faster process.
Okay, got it. So basically, you still like sounds like you get some measurement like some data points, and then you do the calculation. Okay, I see. Yeah, thanks. Yeah.
Thank you. Srikar?
Hi, thanks a lot for the talk. Yeah, I'm Srikar from India. So actually, I had a question about the first part. So you mentioned that you extract different information from, say, different illuminations, or for example, rotation and things like that. Like, is there a way to quantify how much information you're getting from each of these different things, at least like theoretically? I think you showed a graph of, for different cases. I think it was slide 21 or something.
Yeah,
but, like what would be the y-axis of that? Like, how do you quantify the information that you're getting?
Yeah, so this is a slightly expanded version of that slide, so I apologize. It's kind of like a lot on one thing. But basically, we're using a linearized forward model to do this, which is slightly different than our actual reconstruction algorithm, which uses this multiple slice algorithm, which isn't linearized, but they're kind of built on some of a similar basis. But the way that we actually quantified that is we propagate through our system and calculate what our complex transfer function of each image is. And then you can aggregate those into this weight matrix W. So you're seeing these example complex transfer functions here at the top with some pupil coding. And then you can, again, aggregate those to get what your overall like transfer of information of spatial frequency information is in both X and Y, and then also in Z. This is a lot right here, but anyway, this is the general kind of way that we're doing. What you're seeing in these plots here that I showed before was the radial average of this kind of donut-like thing. And so you're seeing kind of these lower spatial frequencies being higher weighted in these coded kind of cases. So that's kind of the method that we used. If you're curious, I can definitely send you the paper that this is based off of out of Leitian's group.
Oh, sure. Yeah, thanks a lot.
Thank you.
There's no one else. I jump in with a question real fast. It might be related to what you were just talking about, but I was wondering, like, you're talking about how the pupil coding can yield like more diverse measurements. And I was just wondering, is there some, like in the measurement space, some mathematical, I guess, description of what makes something a diverse measurement? I'm not sure if that's what you're showing on this slide necessarily.
Yeah, it's a great question. So this is something that I tried to run down for a while. And the thing that I mean, so this ended up being kind of the best description for this particular system and the easiest analysis tool. But the other thing that I was looking at was just kind of an information theory entropy kind of measurement. And that was also somewhat helpful and definitely kind of showed that there's this increase in entropy, but it was a little bit less explanatory as to why in this particular case the low spatial frequencies are showing up more. But that was what ended up being most useful as a more general tool. So yeah.
I see, that makes sense. Well, if there are no other questions, we have 10 minutes left, so maybe we can transition into the advice section.
All right.
Yeah.
Cool. All right, cool. So yeah, I know I've listened to these before and found it very useful to hear kind of what other people have gotten out of graduate school versus postdoc and things like that. And so I thought I'd share some lessons that I learned. Would also love to hear other people's thoughts on this too, because I know it's kind of different for everyone, but also maybe the same. So this is actually a slide that I wrote at the end of graduate school and shared with my group. And at the time it's very unsolicited because I just put it into a group meeting presentation. But now you've actually asked for it. So it's actually solicited thoughts on graduate school. So when I kind of reached the end of my PhD and was looking back at, okay, what did I actually learn from this experience? I realized that I kind of had gained this ability to direct a research project and hopefully communicate about it well. And that this is something that maybe when I came in, I thought I was already supposed to know how to do. But it's not. This is actually a skill that you're acquiring. You're getting trained to do this. And if you knew it when you were coming in, you'd be getting paid a lot more money, right? So I think that's a very comforting thought as a graduate student. You're working on this ability. It's not something that you already know how to do. Another thing was kind of thinking about inverse problems on this kind of larger scale and thinking about how to approach them. You obviously gain a ton of specific skills in, a certain area. For me, it was in optics and algorithms, and knowledge about, specific problems like how do you calibrate out the pupil of an imaging system? How do you align this particular lens well? I mean, it's just like very, very specific information. But even those things can be applicable in lots of places if you kind of abstract it out and think about how that relates to other imaging systems. The other thing that I thought was super valuable was the exposure to lots of really cool research. And I am really happy that I actually went out and tried to listen to research that wasn't just computational imaging. It's super useful to learn about what's going on in your field, but now, especially as a postdoc, that I'm kind of talking to lots of different people and trying to solve different problems, it's just been I've been really grateful to my past self for not just limiting myself to only my research area and looking at lots of different things, because it's amazing the kind of connections that you can then pull through and be like, I know that in physics they're doing this thing. So I think that's really something that I want to continue to do, and I think I would encourage people to do in graduate school also. And then finally, kind of the opportunity to get to know and try to understand the thoughts of a lot of people who are working on really cool stuff, which, I would say this is like your lifelong connections, your friendships. One might even call it networking. I think when I hear networking now, I think more of like just trying to basically make friends with people. And then eventually that might be something that you network. But I don't know, networking feels like a very kind of clinical term. And I think really more it's these kind of relationships and really just that opportunity to understand people and what they're working on. And then also just a lot of self-understanding. So I think graduate school, you're really kind of going through a thing that part of the process, and this relates to that first point again, is it's you are the person that is the product of that whole, the whole process. The point of it is to produce someone who can do research. And so it's not just about, the coding skills that you're acquiring. It's also about like, how do you navigate different situations? How do you kind of value different things in your life, different things like that? So I would definitely say one of the biggest things I learned is that, and would recommend to other people to think about is, trying to look at yourself and understand how you're kind of functioning as you're going through this process, and what you need. So to that point, the other kind of main thing that I kind of came out of and still am kind of refining and thinking about is, how do you find anchor points in your life such that you can do, you know, the cool work that you want to do and have impact? And for me, personally, that's kind of having community and having, you know, friends, family, other kind of community members around me, being able to go into nature and travel, having a team to work with, and then also having fun work with impact. And this is going to look different for everybody, but I think it's very important, not just in a sense of like, you want to be more productive, thus you should have work-life balance, but like, you know, I think you're looking at your life holistically. You want to not just have work be the main aspect, though obviously we're all here, so it's going to be part of what we're doing, right? And then finally, this is just kind of a pet peeve from when I was looking for jobs, but I know a lot of people are also asked the question of, do you want to go into academia or do you want to go into industry? Which we all ask because we've all kind of had to ask ourselves that question at some point. So it becomes this question that, you know, seems to have this overarching power. But definitely when I was going through a job search last year, I was kind of like, I reject this question. I don't like this question. I really want to ask, what's my application space? Because that, I think, more functionally controls what you're doing and where you're working. And maybe, you know, if you're choosing to work in a certain application space, that's definitely more industry related, you might go industry. But if it's something that you can only do as a professor, then you definitely want to go academia. So for me, I kind of mapped out the kind of space of possibilities that I saw for myself at that time. So that ranged from kind of going into consumer products, or maybe more biomedical, which is kind of, it can be a consumer product, or it can be kind of more on a nonprofit scale. Science policy is also something that you can consider. Working in the government can also have very specific imaging problems, which often computational imaging is very useful for. So this was kind of a more useful way for me, at least, to think about career rather than academia or industry, which feels very kind of binary and static. So those are just some thoughts. I don't know if that's helpful to anyone, but it's things that I've kind of thought through as I went along. So thought I would share and would welcome any questions on this as well. So done with our advice section.
Yeah, thank you so much for the advice. I feel probably relevant for people a lot of different stages of their PhD or even afterwards. I just want to check and see if there's any final questions from anybody, either about the technical talk or the advice. Okay, cool. Well, then I think we can probably close off for today. So thank you so much to Regina for giving us an awesome talk and awesome advice. And thank you everyone else for joining, and we hope to see you in the future as well.
Thank you all so much. I really appreciate the opportunity to talk to you.
Thanks so much.